Thank you, Tony. It's actually very nice to be here. I think the last time that I talked in a Mutational Scanning Symposium, we were introducing the technology that the company is based on. So I'm going to show you what we do at AlloX. AlloX is a spin-off of the CRG. Actually, all the co-founders come from the Leonard Lab. And what we're trying to do is map at scale allosteric sites in order to get better drugs to the clinic. And I like to start always with this powerful image. These are the most famous oncoproteins. We know they have been genetically validated. We know their structure, their function, why they cause this disease. And despite all of this rich amount of information, yet few of them are considered druggables. We have FDA-approved drugs that target those targets. And if you see, most of them are the same type of protein, and the few approved drugs that we have are, in fact, orthosteric inhibitors of these kinases, and most of them are not even selective. They cover multiple of these kinases at once. The rest of the proteins in this picture, most of them are not kinases, some they are. But they function, and they are not enzymes, or they function via many other mechanisms. But there's something that it's always mostly in common between them, is that at some point they interact with other proteins to function. And targeting protein-protein interactions is vastly an unaddressed therapeutic space in the clinic. And why is that? Protein-protein interactions are known to be large, flat, and featureless, so it's very difficult to target them with small molecules. And even if we manage to do so, they are highly conserved, so it's very easy that we target, unwanted homologous proteins. It's not impossible, but these are some of the reasons why this has been a difficult task to do. What is an alternative to that? Thanks to nature, we have a possibility, and those are allosteric sites or allosteric switches. And these are regions that are distal from that functional site, in this case, the protein interaction interface, that have the ability to modulate that function, so to modulate binding affinity with another partner. And it could be ideal if these fall into, a cavity or druggable cavity in the surface of the pocket where we can direct small molecules there. And we know for the few allosteric modulators that are now approved and in the clinic that these could be really good drugs because they tend to be highly specific, they can bring selectivity, and this overcomes some of the problems that we face in the clinic, such as toxicity effects. Those makes them ideal candidates or ideal targets for drug development. What's the problem? That for the vast majority of the human proteome, we have no idea where these switches are. In fact, we don't know if the vast majority of the proteome has allosteric switches. And this is because we don't have, or we didn't have until now, a systematic way of mapping this. So what do we need to map these switches? Is ideally we need to massively parallelize or identify biophysics or biophysical properties, so binding affinity between two proteins. And we can do this to identify these allosteric sites via multiple ways. So we can do mutations as perturbations, but mimicking what would be a mutation, a ligand binding, or a post-transcriptional modifications, just to see if this binding between two proteins is reduced. But this binding between two proteins can have two different causes. The first one is, of course, we don't no longer have that binding affinity. And the second cause is because now there's an effect on the stability of one of these proteins, right? We reduce the concentration of this protein, and there's no longer that protein-protein interaction happening. And we know that this is one of the major drivers of protein-protein interaction disruption. So this is the key of the technology that we worked at AlloX that was developed here, that is systematically screening for the effects of mutations on protein abundance and protein binding. And then we use, of course, our favorite technique, DMS, to obtain this in a single pot experiment. After sequencing, we feed all this data into thermodynamical models that allows us to disentangle binding affinity from folding stability. And the outcome of this, and most of the majority of you know very well, is for every single position and for every single amino acid substitution, we obtain that change in binding affinity. So disruption of binding that cannot be explained by changes in abundance. And this is the best resource that we have now if we overlap it with existing three-dimensional structures to identify those allosteric sites, because those are going to be distal from the interaction interface and will have that binding disrupting ability. And this is a very nice case that Chen Chun, from ex-member of the Leonard Lab, ran this technology in the protein KRAS, very nicely identifying that KRAS indeed is highly allosterically active, and very nicely showing that one can identify switches or pockets that ended up being the starting point or the pocket where drugs will be able to modulate or inhibit this particular protein, which we knew for decades that was a major driver of lots of cancers, but until then it was undruggable. Now it's not the case anymore, and we have sotorasib, agarasib, and multiple others that are coming out soon that target this particular switch number 2. So this was a very nice example in a therapeutically relevant protein, and now we took at AlloX, we took this technology, and what we want to do is to be able to identify or map as many proteins as possible in order to identify allosteric switches, and we are lucky, then action some of these switches to develop new molecules. So I'm going to show you a little bit how we think and we operate in this early starting company. So we could start from the human interactome, but that's a vast space. So what we do is reduce a lot and focus on intracellular protein-protein interactions. Intracellular protein interactions are much less accessible to other modalities such as antibodies, so there's where small molecules can really make an impact. And of course, we gather lots of information so that we can say that those two proteins interactions with high confidence are interacting. We apply a lot of other filters, but we try to prioritize for causes of diseases or targets where we know that if we target that interaction, that's going to have an impact, right? That we can make a difference in the clinic, and we can find those patients that would benefit from these solutions. So once we have selected targets, we move them in what we call the pre-platform target validation. The name says it all. We just do very simple checks that the targets and the protein interactions show a good signal on the binding and the abundance assays with its proper controls. And we've done it for quite some. And then we move in what is the map generation, which you already know very well. The last part is actually our goal is to go, and why we started this company outside of the lab, is to go from de-identified allosteric maps and switches into a drug discovery campaign. So I'll explain a little bit of everything, but first I wanted to show a summary of the types of maps and the type of data that we are running our technology on. So we are applying double DPCA and running this technology on various different types of proteins, not focusing on a particular class that are involved in many different pathways. But I wanted to put this together also to highlight that we are getting also very rich and different diverse types of maps. In this case, we originally thought of inhibition allosteric as the one that mainly we would obtain, but we obtain also activation or even maps that tell us about complexes, not only pairwise interactions. So I have a couple of case studies to show here, and the first one is an inhibition map, and in this case, it's on a CDK, a kinase involved in cell cycle progression, where if we inhibit this, we would release, or this could benefit in breast cancer and other modalities. The the interaction partner in this case is the cyclin. That's the regulator that will allow this kinase to be and become in the active state, to start phosphorylating and progress the cell cycle. What was the problem and why did we select this target? It was because so far there are no selective inhibitors of this kinase, and patients have lots of problems and side effects because of that. So allosteric could bring that selectivity, right, which would reduce toxicity because we could boost doses into patients, and potentially we could also overcome resistances, as Matt was mentioning, if those happens on the on-target of the allosteric inhibitors that currently are being used. So at this point, there was no publicly described allosteric sites in this kinase, but we show very nicely that that's not the case. For those that are not familiar with this decay plot, we are showing the mean absolute allosteric potential, so those residues that have the ability to disrupt binding that cannot be explained by changes in the concentration of the protein. And you see that the interface is very much enriched for these binding disruption mutations, but we also find these types of residues far away from the binding interface. And the key question here for the next step is, where are these residues? Do they fall into pockets that we could use as a starting point for a drug discovery campaign? And when I mean pocket, I'm not categorizing allosteric sites as pockets. So an allosteric pocket would be a groove or a cavity in the protein that has druggable characteristics that has allosteric, is enriched in allosteric mutations. And that's exactly the exercise that we do. We take all the annotated pockets or predicted pockets in this particular protein, in this kinase, and we assay which of those are highly enriched for this kind of allosteric mutations, in the same ways that the binding interface is compared to the rest of the surface. And in this case, in this kinase, we identified two candidate allosteric pockets. It was nicely that both have favorable druggability characteristics, and one of them is shown to be drugged in an homologous kinase. So this indicates that potentially this can also happen in this one. I'm going to show a completely different or the opposite map. In this case, it's a GTPase. This is a switchable protein, right, that when it binds GTP can bind its effector. And in this case, this is a GTPase that it's highly frequently mutated in cancer. And when we generated this map, we surprisingly had these very striking blue columns. These are sites. Whenever we mutate, we are actually bringing the two proteins more together. So we increase binding affinity between those two proteins. And very nicely, all the residues that are showing this activating capacity are not located on the binding interface and are acting in an allosteric fashion. What was also nice to see is that when we look at the known recurrently frequently mutated positions, those completely overlap with those activating or classified as activating residues. And now we have even generated a complete map of those activating residues. So the team has also been not only busy in generating as much maps as possible, but also trying to make this process as less cumbersome, faster, cheaper, so that we can get there even faster. So there's a lot of innovation happening at multiple steps, and today I'm sharing one particular focus that we have in the company, that is construct optimization. In this case, we wanted to tackle and work with a DNA binding domain of a FOX transcriptor factor, and the question was always, how do we define the domain, how do we cut it, and how do we put it in when we start analyzing, putting it in our experiments. And initially, our first arbitrary choice had not a very good signal, so we decided to apply this methodology where we use the mutational scanning again, but we are just varying the amino acids at the N-terminus and C-terminus, and we have all these combinations of potential 10 by 10 amino acid different variants. And this we do now with nanopore sequencing so that we can play around with domains of different length. Very nicely, nanopore sequencing and Illumina sequencing correlates much better despite being the input rates and the error rate being much different in nanopore. And the end story of this was that basically just by having two very simple rules, a favorability of amino acids in the N-terminus and another in the C-terminus, could very easily classify or show that we have the best working variants in abundance PCA signal. And just a curious fact, the best working variant differed by one amino acid of our initial not very well selected construct, and that had more than threefold change value. So at least in these kind of fusion constructs, the boundaries of domain really matter, and now we are starting to implement this at the same time that we are validating targets to fit into the platform. And I wanted to spend some time on this part because I think this is at the end goal of this, and I think it's well, this was part of the inspiration to go from the lab and doing DMS to bring something or do a step closer to accelerate and bring better drugs into the clinic. That's a big motivation. So I wanted to share how we are thinking and we are doing it. There's no results I can share yet. This will have to wait for the next Mutational Scanning Symposium, but at least show you how we do it once we have identified those allosteric pockets. So the first thing that we do is what we call a thorough druggability assessment of those pockets. And this implies that we have to identify which is the pocket that not only has that allosteric potential, but has the best characteristics or properties to be drugged. And this involves a variety of different things. It could be looking at the shape and the size, the features of the pocket, but also we can run very shallow virtual screenings or docking to see if that has favorable characteristics to dock molecules, or even play around a little bit with molecular dynamics to see the behavior of the pocket and how it changes on the different states of proteins. The second thing that we focus is on conservation, so trying to see how the sequence or the amino acid content or the shape to this protein is conserved with homologous proteins that probably we want to avoid drugging, bringing those selectivities on those if we drug these new pockets. And then integrating our own allosteric scores or data in trying to identify, key residues that could be a potential starting point or an anchor of those new molecules, trying with the chemistry and the molecule to mimic the effect of the mutation. So once we've done this, then we can prioritize pockets and prioritize the candidate for drug discovery, and we start with virtual screenings. So basically, this consists of docking millions of molecules into the chosen pockets. And of course, we start with different conformations if we feel it's necessary, or we put some constraints so that we ensure that some molecules are interacting with specific selective residues or potential residues that I think could bring that mechanism of action. And of course, AI and deep learning helps speed this process entirely. So at the end of the day, what we have is a bunch of hits or in silico hits, and what we do is select a few of them. In this case, we are talking about hundreds of hits that we are going to do the next step, that is validate them in the lab. And before I forget to mention, this is something that we do. We are slowly incorporating in the company that CompChem capacity of doing those two first initial steps, but we are also working with other CROs or collaborators for doing those additional experiments. And these additional experiments basically is to validate that these molecules are doing what we expect them to do, right? The first one is, do we engage with the target, with that small molecule? That's the first question that we ask. The second is, does this have the same effect as we assay in our experiments, which is, does this disrupt the protein-protein interaction? But then we can go beyond and ask multiple other questions using similar experiments or the same ones. If we are looking for selectivity testing with homologous and seeing that there's a differential potency with different targets, with cellular assays or biochemistry assays, we can go beyond that protein interaction and see how these affect protein activity or downstream effects on the pathways that are known as biomarkers and so on. And this was just, we thought that this is, I would say, an alternative approach, but I think it can be also complementary, but a different way on how we could do, bring new molecules or identify allosteric modulators compared to more traditional high throughput small molecule screenings that have been done traditionally. The benefits that this bring is that it's a very unbiased, high throughput, comprehensive approach, right? The problem is that we don't start with that initial chemical matter, but we really limit and focus. We do this rational structure-based drug design that will allow us to limit the size of those libraries to test, right? We're talking about hundreds rather than millions of compounds. Screening millions of compounds, it's not easy, it's not cheap, and it takes a lot of time. And not only that, there's other problems. We have biases, either because we have biases towards functional orthosteric sites or because the libraries themselves, the compounds are actually biased towards certain active sites. And that said, that's how we think that DMS, such a DMS allosteric mapping approach, could impact drug development. Not only starting or having this initial very focused and narrow way of focusing on a specific pocket to start a chemistry campaign, but actually complementing the more traditional or chemistry first approaches. There's a lot of times where there's these massive screenings where you know that you have binders, but those binders still don't show functionality, and you know that they are not binding on the active site. So using this kind of information and overlapping these two sources of information could help prioritize which chemistry needs to be tuned even further with medicinal chemistry to get to that functional outcome. And with that said, just gonna thank the people that made this possible. This is the most recent picture. Actually, this was taken about 2 weeks ago because we just moved. We were born at the PRBB, very proud of that, and very sad that we left, but happy that we now have a new lab where we can grow even more. So thank you all for listening, and happy to take any questions.
Thank you very much, Julia.